Alright, we are going to do some review notes to help us focus a little bit more on what we did during the diffusion through a membrane lab, and also to kind of pull all of our pieces together when it comes to cell transport and how things move in and out of cells. So you're going to need this note sheet that you guys got before. It was on a full page like this, but we are going to cut it out. So you're going to cut along the black line and then cut the black lines up the middle. So it's going to look like this. And then we're going to attach this to the blue sheet. You can use the one we used on our previous notes, or you can get a new sheet if you need it. We're just going to take a little bit of glue just along this little top section and stick it right down to the page like that. So we're going to be able to put some notes underneath. We also want to label this so we know what this is related to. So along the top of the page, you want to label this diffusion through a membrane. Lab review notes. Okay? Alright. If you need to, pause the video, get yourself all set up, and then make sure you have some colored pencils or markers ready so we can label our diagrams and fill in some notes. Alright, so we're going to start with labeling the parts of the model that we have in these pictures. So this should look really familiar, very similar to what we did in lab. You do want to just ignore the little labels on here because they don't quite match what we did. So don't even worry about those. We're going to add our own labels. So we're going to start with the baggie that we have here. So find yourself a green color pencil marker, or it doesn't matter. And we're just going to go around the outside of this baggie. And you want to do this in each of your pictures. And what this green that we're shading in represents, this is the cell membrane. That's what the model is trying to show us. This whole thing is a cell. And then the baggie itself is the cell membrane, right? We had inside of this cell two different materials. So if you remember, we had starch. So we had starch inside, and we also had a glucose solution inside, right? Glucose and starch. So we may want to make ourselves a little note below down here. So S equals starch and G equals glucose. Starch is a small, oops, no it's not. I apologize. Starch is a large molecule, and glucose is a small molecule. And these are both carbohydrates that need to move in and out of cells. So starch, large molecule, glucose, small molecule. Okay? And we had starch and glucose inside of our cell, and then we had this environment outside of the cell. So we had water inside the beaker, and in that water we also had iodine, right? So our iodine, grab like an orange if you can find it. And remember, our iodine was sort of an orangey brown color, and we put that in the outside water environment. So I equals iodine, and that is a starch indicator. Starch indicator. So it tells us if starch is present. So here's our first setup. We have starch and glucose inside of the cell. We have iodine in water outside of the cell, right? So this is our starting situation. So let's actually describe that underneath here. So we're going to flip up our first page. And we had a baggie with starch solution and glucose solution. And we placed baggie in water and added iodine to water. Started amber brown. Right? So we had the baggie with starch solution and glucose solution, put the baggie in water and added iodine to the water, and it started out an amber or brown color. Right? So let's actually shade that in so we should know what color this water started out. So you can get like a brown. I'm just going to add a little kind of orangey red to that, I think. It's a good iodine color. Okay, so that was our start. After it sat for a little while, let's go on to what happened after it sat for a little while. So let's start with what we saw as far as color. So the water was still kind of colored, right? It certainly didn't look too dramatically different. And since we could still kind of see the color in the water, that tells us there was still iodine present. Let's think about what it looked like inside of the cell. We started to see sort of some purpley black inside of here, right? It's like a very dark purpley black color. So that tells us that iodine moved into the cell. How I know that is because iodine turns purpley black when it's in the presence of starch. So iodine must have moved into the cell. I now have iodine in here. And that tells us that I also still had starch because my indicator reacted with starch. So it still has to be starch in here. And we know from when we tested this with the heat test with the glucose indicator that there was still glucose inside because there's no glucose outside. Alright, so still have glucose and starch inside the cell. Still have some iodine outside of the cell. Alright. So let's describe that. After it sat for a while, so iodine diffused into the baggie. You could tell because the baggie contents, changed color. Purple indicates the presence of starch plus iodine. All right. If we had let this sit, or I think I even showed you guys, after we let this sit overnight or for a really extended period of time, what you will see happen is this whole baggie is going to start looking purpley black. And the water is basically going to look clear, like regular water. And the reason for that is I have my glucose in here, and I have my starch in here, and I have my iodine. So there's no iodine left outside of the cell. It has all diffused in. So what this tells us is that the starch molecules are unable to diffuse out of the cell, but iodine molecules can diffuse in. Awesome. We're good.